friends, welcome to eStudent. In this video, we will be discussing Iit Jam Biotechnology 2019 question paper. Friends, we have also discussed 2019 and 20 question paper which is available in our channel. So please go and visit. Now we will be discussing the section A. So the first question, which one of the following protozoan parasites belongs to the phylum Epicomplexa? Toxoplasma gonidie, Leishmania donovani, Entamoeba histolytica, Trichomonas vaginalis. So friends, we know that all of these four are the protozoans, but they are asking which one of the protozoan parasites belong to the phylum Epicomplexa. So let us see. The Here we are seeing that this is the Epicomplexa phylum, and under this class is Conidacida, subclass is Coccidacina, order is Eucodiorida, family is Sarcosistidae, genus is Toxoplasma. So here we can see that the protozoan parasites belonging to the phylum Epicomplexa is the Toxoplasma gonidie. Okay, so coming to the next question. Which one of the following statement is correct for mycoplasma? So we have to know first a few things about mycoplasma. First, mycoplasma is a gram-negative bacteria. Okay, these bacteria are different from other bacteria because these lack cell wall. Okay, other bacterias, gram-negative bacteria, they have the cell wall, whereas the mycoplasma lack the cell wall. So which one of the following statements is correct? They have asked in the question. So their cell walls are of definite shape. This is absolutely wrong because mycoplasma never have the definite shape. They are pleomorphic. So what is pleomorphic? They keep on changing their shapes. They can like change their shape. At one time they are at spherical shape. Sometimes they are in another shape. So they are not of different defined shape. They are resistant to lysis by osmotic shock. So this is also a wrong option because they are uh, not see they are actually resistant to lysis. They are actually telling about the plasmolysis. Okay, so these are not resistant to plasmolysis. Okay, so actually they are not resistant, but they are given in the question that they are resistant to lysis by osmotic shock. Okay, so this is a wrong option. Their growth is not inhibited by penicillin. So what does penicillin do? Penicillin actually inhibits the growth of cell wall. Okay, and if the cell wall synthesis is inhibited, the cell growth is also then inhibited. Okay, but in the case of mycoplasma, we are not even having the cell wall. So how will the growth be inhibited? So their growth is not inhibited by penicillin. This is a correct option. Okay, they are non-pathogenic to human. This is absolutely wrong because mycoplasma is actually pathogenic to human. They mycoplasma pneumoniae. They are actually pathogenic to human. Okay, coming to the next question. Which one of the following organelles is enclosed by a single membrane? So, first we will cut off the options which are absolutely like irrelevant here. We know we are studying it from our childhood that mitochondria is a double membrane structure. It is having outer membrane and an inner membrane. So this is the wrong option. Here ribosome is not even having a membrane. Okay, so here endoplasmic reticulum and centrosome, the correct answer is endoplasmic reticulum because it is only having a single membrane. Okay, Coming to the next question. Pyramid of energy in the forest ecosystem is. So this is the pyramid of ecosystem. So whenever there is, we are seeing the pyramid, it is always an upright structure. Why? Because you see there is always a decrease in the energy here. You see the sizes are always here decreasing. Okay, this is having the highest energy, this is having the less energy when compared to this one, and again this one is having less energy when compared to the primary consumer, and secondary consumer is getting the less energy when compared to the primary consumer, and tertiary consumer will get the lowest energy from all of these. So the pyramid of energy is always upright. So this is a question given from like 11th and 12th class. Okay, very easy question this one is. We always know the pyramid of energy is always upright. Okay, coming to the next question. In the feedback regulation of an enzyme, the end product binds to. So the end product in the feedback regulation of an enzyme will bind to active site of the enzyme. No, it will, the substrate is going to bind to the active site of the enzyme. um, Enzyme substrate complex? No. It will bind to the substrate? No. It will always bind to the allosteric site of the enzyme. So B is the correct option here. Coming to the next question. What is the source of electrons in photosynthesis? So we know that whenever there is a light on photosystem 2, the electrons are excited from photosystem 2 to the P2 complex, that is plastoquinone. Here the source of electrons here is the water, H2O. So here the electrons from the water gets excited and they are transported to the plastoquinone. So the correct option is B. This is also a very easy question. We know that the main source of electrons in the photosynthesis is water. Coming to the next question. Viral capsids are made up of morphological subunits called capsomeres. One of the common capsomeres is icosahedral. The icosahedron is a regular polyhedron with. So this, in this question we cannot apply any logic or something. These is a like a purely question that we have to remember these things. Okay, so this is the, virus. Okay, so this is the icosahedral capsid of the virus. So it will always have 20 triangular facets and 12 vertices. Okay, so here B is the correct option. Next question. Which one of the following features should be present in a protein to generate strong immune response? Antibody production in an animal. So here what they are asking, which response is very important to produce the antibody production. So we know who produces the antibody in the cell. The B cells produces the antibody. So what happens here, the antigen will come and bind to the antigen presenting cells. Okay, so they come and bind with the antigen presenting cells. So these antigen presenting cells can be B cells, dendritic cells. Okay, these cells can also be the macrophages. Okay, so if this antigen is having an epitope for this B cell, then only this antigen can come and bind here. Okay, so this must have an epitope for B cell. So at least one B cell epitope must be there. Okay, now this, when this antigen is engulfed here inside the cell, these antigen is usually fragmented, matlab they're cleaved, and then there one one of the antigen fragment is presented on these B cells, so that they can be recognized by the T cells here, T helper cells, so that these T helper cells can recognize this antigen. Okay, so there is a this is a T cell epitope. Okay, so the epitope, why it is called a T cell epitope? Because this T cell is going to recognize this site. Okay, so here because this is cleaved, this antigen is cleaved here, this is called as proteolytic cleavage. Okay, so here it is undergoing the proteolytic cleavage, and it is also having at least one T cell epitope. Okay, so the correct option is D, 1, 2 and the 3. Next question. Match the entries in group 1 with that in the group 2. Okay, so here they are given about toxins. Okay, here we can see everywhere there is the mentioning of toxin. So first we'll understand there are basically two types of toxins. What is endotoxin and one is enterotoxin. So I'm so sorry, it is endotoxin and exotoxin. Exotoxin. Okay, so endotoxins will always like these are associated with the gram negative bacteria. And what do gram negative bacteria in special have? They have the lipopolysaccharides. Okay, these are associated with the lipopolysaccharides, where exotoxin, these are involved with the gram positive and negative bacteria. Okay, so lipopolysaccharides, we know they are endotoxins. So endotoxin, let's match this. And tetanus toxin are involved in they are the neurotoxins. Okay, and the cholera toxin is an enterotoxin. What are enterotoxin? These are associated with the intestinal cells. Okay, they affect the intestinal cells. So in cholera, the intestinal cells are actually affected. So they are enterotoxin. They affect in the intestine. And the left one is diphtheria toxin, which is a cytotoxin. These affect the tissues of the cell. Okay, so P is 3, which is given here and here. Q is 4. So here we are having 4. So C is the correct option here. Okay, so let's go to the next question now. Protoenzyme pepsinogen is secreted from P. So what they are giving? P is secreting pepsinogen of gastric mucosa and converted into active enzyme pepsin on exposure to Q. So pepsinogen is getting converted into pepsin by the exposure to Q. So Q is converting the pepsinogen to pepsin. Okay, um, secreted from R. So this Q is secreted from R. Choose the correct combinations. Okay, so we know that pepsinogen is secreted from the chief cells. Okay, they are present in the stomach, which are secreted from the chief cells, and the pepsinogen is converted into pepsin with the help of hydrochloric acid. Okay, and the who secretes this hydrochloric acid? These are oxyntic cells. Okay, so here the correct option will be the P cells releases, this chief cells releases the pepsinogen. This pepsinogen is converted into pepsin with the help of hydrochloric acid, which is secreted by oxyntic cells. Okay, so A is the correct option here. Coming to the next question now. When bacteria are grown in glucose depleted media containing the high concentration of lactose, expression of lac-operon genes is activated by. So whenever there is an option given glucose depleted, we must always have in our mind that in this situation the repressor will not be bound to the operator sequence. So this is our X, Y and A genes present here on the operon. Here is a operator and here is a promoter side and here is a regulator side. This is a regulator side. Okay, so this regulator will produce the repressor. So whenever there is a lot of glucose present, this repressor is always bound to this. This is repressor. Okay, repressor is bound to the operator so that the RNA polymerase cannot go and transcribe these genes here because glucose is already present. So we don't want to produce more glucose from the lactose present. Okay, so but here condition is given glucose is depleted, glucose is not there, so it has to be produced. So from what it is produced? It is produced from the lactose. So first the repressor has to be removed from the operator so that the RNA polymerase can go and transcribe these genes and use the lactose to produce glucose and galactose. Okay, so let's see the option. The binding of lac repressor in the operator site and see lac repressor is binding in the operator, so these genes will not be transcribed. So the answer is clear from here on that this is a wrong option. Okay, the dissociation of bound lac operon, yes, this is correct. From the operator side, yes, and binding of CAMP cap complex in the cap site. So, okay, I haven't explained this cap and camp. So, whenever there is a glucose present here, just beside of this operator and, okay, I'll explain with another color. Operator and promoter, just beside this, we are having a cap and to this cap, camp will come and bind. When usually when glucose is present, this is not there, this complex is in very low concentration, camp. But whenever there is glucose level totally goes down, this cap and camp complex, so the camp complex will increase in number and this will increase the efficiency of RNA polymerase getting bind with the promoter site and uh, going for the transcription further. Okay, so the dissociation of bound repressor, so this repressor is dissociated from the operator side, binding of cap complex with the cap site. Okay, so cap site, the camp will come and bind here and this will increase the efficiency of RNA polymerase binding to the promoter site and then transcribing this. So this option is the correct option. So next options also, let us see the dissociation of bound lac or repressor only from the operator side. Okay, only from the operator side. No, because this has to be uh, this has to be removed, but here we must also include that cap has to bind to the cap complex. Only here it is mentioned, so this is a wrong option. The dissociation of both lac repressor and the operator from operator side and cap complex from the cap side. No, this is the wrong option, because this should not be removed. This should not be dissociated. This should actually be included. Okay, so this is a wrong option. So the correct option is the B option. Now coming to the next question. uh, Match the hormones in group 1 with the functions in group 2. So this question is also a purely like remembering the functions of the hormones, the whatever they are secreted from which of the glands, and those questions these are. Aldosterone, so we know this helps in the reabsorption of Na plus and water from the kidney. So aldosterone is 2. Luteinizing hormone, we know it stimulates the synthesis and secretion of aldosterone from the testes. Arterial natriuretic factor, it causes the dilation of blood vessels and reducing the blood pressure. And epinephrine, we know that epinephrine always increases the heart rate. So here is the 3. So P2 it is given here, and Q is 1 here. So Q is 1 here. So this is the B is the correct option. Yeah. Let's go to the next question now. Match the entries in group 1 with that of group 2. Okay. So here this is a very important part. Vitamins always is given in the question paper of IIT Jam. So it's a very important topic to remember. So vitamin B1 is involved with the thiamine pyrophosphate. Okay, vitamin B5 is the coenzyme is coenzyme A here. So here vitamin B2 is also known as the flavin. Okay, so riboflavin. So here flavin mononucleotide. Okay, so with these options we can even if we know about like one vitamin, we can answer the whole question. Like vitamin B5 here, B5 is involved with the coenzyme A here. So you see R1 is only given in this question. So D is the correct option here. Okay, and see B1 is involved with the thiamine pyrophosphate. Vitamin B2 is involved with flavin. Vitamin B5 is involved with coenzyme A. Vitamin B6 is pyridoxal phosphate. So D is the correct option here. This is the 13th question. Coming to the next question, it's the 14th question. Match the entries in group 1 with those in group 2. So proline, we know proline is a cyclic amino acid. Okay, so proline goes to 2. Oxytocin, we know it is an hormone, so peptide hormone here. Aspartame is a artificial sweetener, and penicillin is beta lactam. So P2 and Q4 is given in this question, so E is the correct option. So by this we complete our 2019 section A discussion, and in the next videos we will be discussing the section B and section C. So thank you, hope you have understood, and if you are having any doubts, please ask in the comment box. Thank you.